Okay, so thank you all for joining our webinar today. My name's Sarah Chowdhury. I know a lot of you have joined today, so hi to everyone out there and hope you're all doing well. Also online today we have Courtney, who's over in the US. We also have Sabrina and Boris, who are part of the Gyger team in Switzerland. And it's with great pleasure to introduce Helen Plant also, who will be presenting this webinar today. So Helen is the UK team leader for automation. She's based up in Alderley Park in the UK. We've known Helen and the team for many years, since 2015, and over the past 6 years we've been working in close collaboration with Helen and the team. During our collaborations we've enhanced the flexibility of the CERTUS FLEX, and it's also enabled us to get a closer look and an insight into actually what is needed onto the HTS workflows. So let's go straight over to Helen, so she can tell us in more detail about the CERTUS FLEX and how it's implemented in the HTS workflows. Thank you very much.
Thanks, Sarah, and thank you for the invite to present today. So yeah, I'm based over in the UK, in Alderley Park, and I head up the automation team within the HTS group over here. And we've worked, as Sarah's mentioned, we've really tried to, we've had the CERTUS FLEX for several years, and I'm going to talk to you today about some pieces of work that we've done to really optimise that device to enable us to use it as a high throughput screening piece of kit. So just a little bit of background about AstraZeneca. We're a global company with around 64,000 employees. Annual revenue is about 22 million, and of that 6 billion is invested in R&D. And R&D is centred in several sites across the globe. Within AstraZeneca, we have the Discovery Sciences function, where AZ has actually centralised all their early phase preclinical research. And within Discovery Sciences, we have various departments, including hit discovery, our sample management group, assay development, secondary screening, and reagent generation. And I sit within hit discovery. And hit discovery is where we've actually centralised all our hit finding modalities. And as you can see, that's where the high throughput screening group sits. So in terms of high throughput screening, what are the challenges? What are the requirements for equipment that we use? There's a large range of commercial equipment available for screening, and really for us, not only is accuracy important, but speed of dispense, speed of the step. Within the group, we use equipment either standalone, or we also integrate it onto our more complex automation platforms. What we found over the last decade really is that although the equipment's there, it's had a reduced focus for vendors on high throughput screening. The number of screening groups has reduced over the few years. There's many more academic groups, and maybe they don't have the budget for an HTS minimal piece of kit. So we took that opportunity really to collaboratively work with vendors such as Gyger to help drive development of products towards the HTS environment. And these have covered both hardware solutions and software enhancements. So the CERTUS FLEX Liquid Dispenser, for those of you who don't know, just to give a quick overview, obviously it's a microplate dispenser, and it uses that microvalve technology. And importantly for us, that is contactless dispenser, so we don't have any problems with potential carryover issues. The microvalves are for us very precise and accurate. They dispense over a wide range of volumes, which makes it very amenable for different types of assays, and can handle a range of viscosity fluids. But also importantly for us, it can be integrated onto automation. So the first part of my talk, I'm going to talk about the collaborative work that we've done with Gyger to develop the Flex for HTS. And the first area that I'm going to talk about is the speed of dispensing. So when we first took the dispenser into our labs, we did some timing work with it, and shown here is the head of the Flex with valves in positions 1, 2, 3, 4. And when we used the dispenser just with a valve in position 1, we did some timings of how long it took to dispense to a 1536 well plate, which is our standard plate type that we use in the HTS lab. And you can see if we're dispensing either 1 or 5 microliters in the blue, that if we're using only 1 valve per reagent, the time to dispense is at over 3 minutes a plate for a microliter. Software on the Flex actually allows you to use multiple valves to dispense a single reagent, and we choose to use at the moment up to 4 microvalves. And you can see if we do that, in terms of the time to dispense. The orange is using 2 and the grey is 4. If you use 4 valves per reagent, you can actually bring the dispense time for a whole plate for a microliter, which is kind of standard volume 1536, to less than a minute. So that's great. That reduces the addition time per plate to by 75%, which brings it into timings for how we would the timelines for HTS primary screening. So yes, we can use up to 4 multiple valves to add a single reagent. And actually shown here in the top heat map is some data from a biochemical assay where we've just got the percent effect data. And you can see actually we're using valves in position 1, 2, 3 and 4 to add the same reagent. The position in valve in position 1 is actually dispensing a slightly different volume to valves 2, 3 and 4. We use standard factory calibrations for the valves. We don't choose to custom calibrate it because we're only dispensing 1 volume to the whole microtiter plate. So we actually use Gyger software to apply a correction factor, which we can determine gravimetrically to adjust slightly the volumes that each of these individual valves is dispensing to, we call that being balanced. So in the top, you can see we're using non-balanced valves, so we've not got that correction factor applied. You can see 11% CV, whereas when we've actually been able to apply those valves, apply the correction factors to those valves, you can see the heat map, the pattern has disappeared, and CV values are significantly reduced. I will say for this though, you have to apply a correction factor, it's only for a specific volume. So again, that makes it amenable for HTS purposes. If you need to use it for adding different volumes through the same valve, you would need to do a custom calibration. We also have some data just shown down here where we've got a cell-based imaging assay where we're showing that either using 4 valves or 1 valve on the y-axis to add the same reagent, that we don't particularly see, we see pretty good correlation of data. So we're confident that we can really speed up our dispense times by using multiple valves for a single reagent. The next area that we looked at was the ability of the CERTUS FLEX to see whether it could dispense more than 1 reagent at the same time. The first version of software could actually only do 1 reagent, and that would obviously require us to use at least 2 or more dispensers per HTS assay. So we worked with Gyger to develop a version 2 of the software, which actually supported that simultaneous dual or more than 2 multiple reagents can be added at the same time. And again, using that multiplicity of valve additions, so you've got multiple valves adding different reagents across the plates. In terms of timing again, so the version 1 software, I've just shown some data down here, 1536 well plate where we're using 4 valves per reagent. We obviously read 2 CERTUSes, adding a microliter per well on each of the different CERTUSes takes just under 2 minutes. Whereas in version 2, again using 4 valves reagent, we can combine the addition times on 1 CERTUS, so efficiency gains there, and we can bring that timing down to less than a minute a plate. So just show you in the middle actually how that's set up. So we've got potentially first 4 valves adding the enzyme, and the second 4 valves adding in a substrate, and then the microtiter plate as it passes under the head. Both reagents are added pretty much simultaneously. And on the far right, we've just got a graph of some data, again, a biochemical assay here. We've either chosen to add the enzyme and substrate separately on 2 different CERTUSes or added together. And you can see in terms of the correlation for the IC50 data that we see very good agreement and very good assay stats for that particular assay. The next area I'm going to talk about is actually a hardware development that we did with Gyger. So I think as you've seen at the start of the presentation, the CERTUS FLEX originally was provided with an 8-channel straight head, as I'm highlighting here in my pointer in the top right of the picture. We know that if you use quite high-speed dispensing straight into the well, in terms of biochemical assays or any reagent that's got high protein or detergent concentration, this can result in the bubbles or frothing. And the way that we've resolved that is we would actually centrifuge the plates post-addition. The other area where straight dispensing can cause these issues, we found that if you've got cell assays with monolayers, that actually can disrupt the monolayer by sort of damaging it as the dispense reagent hits, and that can introduce assay variability. So we worked with Gyger to see if we could actually modify develop a new dispense head with actually dispense at an angle down the side of a microtiter plate down, so actually much more gentle, and you can see here as shown in this image here. Giga developed a sort of prototype number of heads, either 25, 20 or 10 degree angle, and they brought those to the labs, and we tested just dispensing, shown here, a 1% detergent solution, and you can see a photograph of the microtiter plate, the straight angle head. You can see clearly bubbles in the bottom 2 rows, but introducing an angle eliminates those. So these plates haven't been centrifuged, and you can see no bubble formation. We did some more work in terms of actually looking at assays as well, and so shown down here in the bottom right is some biochemical assay data, either using the straight head with no centrifuge step afterwards after dispensing. You can see we've got pretty good CVs, but actually if we introduce an angled head and again no centrifuge step, you can see quite a significant reduction in the percent CV, so improving that data quality. And just shown in the top right of the picture, the actual standard product now that Giga supply is actually a 22 degree angled head. They identified that that was the optimum angle to use, and you can now buy either a 4-channel angled head or an 8-channel angled head. These modified dispenser heads have really helped us both improve our phenotypic assay quality, but also serendipitously we're also able to simplify our biochemical assay protocol. So a gain there for us really. We've also continued over the years with Giga. They've been very receptive if we've come up with suggestions for other parts of kit that are associated with the Flex. So we've developed a number of peripherals with them. We've developed a universal bottle holder, which can hold bottle sizes from either a 50ml Falcon up to 500ml bottles. Can fit all sizes in them and hold them securely, again combined with the air pressure adapter for the Flex itself. With an HTS we use large volumes and large numbers of plates, so actually if you add into 100 or 150 1536 well plates, actually you need quite a large amount of reagent volume. So Giga developed some larger 500ml conical B-based bottomed bottles with the ability to do multiple 1 in, either 4 out or 8 out reagent dispensing. So again, making it really amenable to HTS. These are now catalogue items that are available as a standard product. So we've really helped with working with Giga to make that very flexible product to meet our needs. And just to highlight the last piece of development work that we've been doing recently, this is now a product that's just come to market. We worked with Giga to develop an offline cleaning station for the microvalves. So housekeeping of your microvalve is incredibly important for maintaining the accurate dispensing. And obviously if dispensing through reagents for hundreds of plates, there's potential for blockages, or you really have to keep them very clean. So you can clean those manually, and Giga had a product on the market for manual cleaning for several years. But we'd identified an opportunity to actually have a sort of plug and play. Users could put their microvalves into the automated cleaning station, press a button, walk away, come back half an hour, and their valves are clean. So the actual controller unit is shown here in the picture. You can program that with different wash programs. You can use various different cleaning solutions which are pumped through. These then pass through the microvalves, which are shown here in this close-up of the microvalve holder. This can hold up to 8 microvalves, and then there's a waste bottle attached. This microvalve holder can actually sit within a sonicating water bath, and we've actually used it with a heated sonicating water bath as well to just physically start to disrupt dislodge and remove any internal debris while wash solutions are flowed over them. And we've had quite a lot of success with valves that have become, they're used in a HTS campaign, they can become quite used and dirty, and we would like to keep them clean. We reuse our valves for multiple HTS campaigns. So we've been able to establish a very robust cleaning methodology with an 8-fold time saving. And again, that ability to plug the valves in, walk away and leave them, the users are really liking that. So for the second part of my talk, I'm just going to talk about how we've actually automated the CERTUS FLEX. So we work with High-Res Biosolutions, who are our automation provider, to automate the Flex on the CoLAB Flex carts. And shown here is a dispenser cart. There's different flavours of the Flex carts, which I'll show you in later slides. But just to show here, this is a dispenser cart where we've got central precise A-cell robot sitting on the base of a cart, which is on wheels, so it's a modular and can be moved around the lab very easily. It's got a UPS, so you don't need to power everything down as you move the cart around. You can see it's very equipment dense around the amphitheatre of the precise robot, and it's quite rich in terms of the vertical space of the cart. And shown here, we have two CERTUS FLEX per dispenser cart, along with a multi-drop combi and a blue cat centrifugal plate washer and some ambient plate storage. In terms of configuring our automation, we have obviously the Flex cart itself can run on its own. It's got its own robot, so it can do some simple plate processing steps. We can also bring the carts together on a cart-to-cart platform where carts are docked together, and one precise robot can hand over plates to the other precise robot. So bringing together, for example, here a dispense cart with a multi-mode reader cart, you can run. We run simple biochemical assays at full 1.8 million HTS campaigns on our cart-to-cart platforms. And for more complex assays, we can actually dock the carts around a 6-sided microstar platform, so which then the KUKA central robot can feed handovers to each of the individual carts around the system. So that's for the much more complex assays. And that modularity and flexibility allows us to reconfigure our platforms very quickly. So just a little bit more detail. As I mentioned, they're very compact, each cart. A key for us is actually we don't have any shielding for our robots, both the KUKA and the precise robot. We've worked with High-Res to meet safety standards in the UK, so actually we can have open access, which the users really like. Each cart is connected, has its own instance of Solario and it connects to a central database, so it's very flexible in terms of equipment and assay mobility. Just to highlight, on the right hand side is a range of assays that we can screen within the HTS lab, just a few examples. And I've highlighted where the Flex cart with the CERTUS FLEX is really pivotal to these different assays. So as I mentioned, you can run the Flex cart on its own for a simple cell plating or cell staining protocol. You can combine for a simple biochemical assay with a multi-mode reader, for example. And then as we've moved on complexity, for example, as we move towards more high content imaging assays, you can see we have a whole range of different Flex carts that plug into the KUKA microstar platform. So we can really scale our automation to make it versatile, both to the assay format and to the library size that we wish to screen in the HTS group. Sticking with the automation, we also, just to highlight some work we've done to actually automate the cleaning of the CERTUS FLEX. As I mentioned earlier, cleaning of the microvalves is really important for high quality data. And we actually use, on each dispense cart, we have Vici valves, just shown here. So the tubing of the CERTUS FLEX actually goes through a Vici valve, which can switch positions. We have up to 10 positions that the Vici valve can move to, and this just accesses then rather than from a reagent, maybe in position 1, you might have wash solutions in position 2, 3, 4, and 4, etc. And within High-Res and within our software, we've actually we have condensed protocols, subroutines for the CERTUS and other devices that we can clean, that you can actually use scripting to switch to the different valves, prime through different reagents or wash solutions. And these have been iconized into small subroutines to really help make it simpler to use and for easier error recovery for the users. So just to summarise how we use the CERTUS FLEX now in HTS with the modifications, you can see we now have the 8-channel angled head on all our CERTUSes. We use the version 2 software, which can allow us to dispense multiple reagents at the same time, and we routinely use more than 1 valve per reagent to get that speed of dispense time. We always aiming to have less than a minute per 1536 well plate. And the use of automated dispenser cleaning has really allowed us to enable out of hours running of the system, but knowing that the system, when we come into it the next day, that the device is ready to run again. We haven't got to spend a lot of time cleaning it. So switch my pointer off. So I've just got a video here I'm going to show you. I thought it might be interesting just to see how the actual carts move with the CERTUS FLEX as our focus. So here we've got a dispenser cart and a reader cart paired up on a cart-to-cart platform. You can see here the precise robot is going to access a microtiter plate which has got a lid on from our random plate storage. So it takes the plate and lid. We use a lid valet to suck with air suction to remove the lid. Plate is then barcode scanned and placed onto the CERTUS. And for this, you can actually see the CERTUS head passes over twice. The first time is to add buffer controls to certain wells. And then you'll see we've actually go back and use, this is now adding enzyme and substrate to the actual assay wells. You can see here that the precise robot has now gone to pick the next plate to hand over. I'll show to demonstrate the handover onto the reader cart. So it's popping its lid back on, regripping to hand it over in the correct orientation. And now the precise robot on the reader cart will come and access that plate, place it into its random storage location. While the second plate is just about to finish dispensing on the CERTUS. So at that point I'll finish. I'd just like to acknowledge all my colleagues within the AstraZeneca UHTS group. This is obviously a large amount of their work, and I'm showcasing it for them. I'd like to acknowledge the guys at Gyger who we've had a great working relationship over the years, and also to acknowledge High-Res Biosolutions and CBRA. Okay, thank you very much.
Thank you so much for that. That was a great presentation, Helen. Thoroughly enjoyed your presentation and learnt again so much more. And I hope everybody else found the presentation very useful. Also, the video at the end was just perfect. We do have a few questions, Helen, so we're just going to go straight into them. The first question is from a friend of everyone's, Nicholas at Galapagos. He's asking, do you apply the correction factor for all types of solutions, and how much time does it take?
So we use all our reagents that we put through the CERTUS we regard as being basically water. So we everything's an aqueous or a water-based solution in terms of the factory calibration that we use. We do apply it we actually check and verify our correction factors before every run. And routinely, for example, if we've got 8 microvalves, it probably takes a person maybe half an hour to verify the valves before we start off the run every morning. We do choose to do this. We found it is beneficial in terms of data quality. We actually, because we're running the number of plates we run per batch, we'd rather spend half an hour and verify and check our correction factors before we start off. They do change slightly, but not significantly, but we like to just check that that's happening. We're also actually working with High-Res at the moment to see if we can automate that valve calculation. We have a plate weigh on the Flex cart where we can actually weigh a plate which has enough sensitivity compared to a balance on the bench. So that's work in progress, but we're hoping to automate that, so users could just load on some microtighter plates and come back half an hour and their valves have been calculated for them. They still have to go physically into the CERTUS software and apply them, but hopefully there should be some time savings there.
Thank you. I hope that's answered your question, Nicholas. We also have another question from Karen at PPSC, which we also know as well. Helen, do you clean your tubings or only the valves with the Vici valves?
So that's a good question. So the Vici valves actually only clean through the head. So we are aware that there's kind of a leg, the bit between from the bottle to the head itself will still have reagent. And so the users, when they come in the morning, are aware that they still have to kind of flush that through with water before they would start loading on new reagents. We'd love the CERTUS to be able to empty back, but obviously that's not an option at the moment. So.
Okay. We also have another question about the valves from Rick. How long do your valves last for?
Wow. So I would say we run, in terms of a HTS campaign, a 1536 well campaign is adding to probably around 1500 microtighter plates, and we probably will use our valves for at least 3 campaigns. So you can see the number of dispensers that we're doing. It is assay dependent. Obviously, some reagents we've had more issues with, and they potentially cause blockages or have been difficult to clean. That's more down to the reagent, but for standard more aqueous-based protein enzyme substrate reagents, we reuse those multiple times.
Okay, thank you. A question for the Gyger team from Nicholas again. When will the cleaning station be commercially available? So we can answer that for you. Nicholas, if you want one, you can have one now. Helen has already ordered hers, so we haven't officially launched it just because we wanted to do it at FLAS in the new year. However, we won't, you know, it's not going to be possible. But if you want further information about ordering, please get in touch with us. We will officially do a launch in the new year, and but it is actually available now if you are interested. Hope that's okay, Nicholas. I'll wait for your purchase order. Do we have any more questions from you, Courtney? Oh, here we go.
I was trying to paraphrase some of the answers so that they were typed in based on what Helen was saying. So no, I think we have a few more questions coming in at the moment.
Okay, we have another one. Using single and or multi-valve dispensing, do you observe priming effects, Helen?
We always prime our reagents before we start off. So users will physically go and load their reagents onto the CERTUS and check it's all primed through. And we routinely, actually, we generally would run what we call a dummy plate or a waste plate as part of our screen. Obviously, if you're running 100 plates, running one dummy plate isn't particularly wasteful. For smaller numbers of plates, that could be impactful. So we don't tend to see particular issues with any lead-in or priming effects because we don't actually go and look at the dummy plate data. I suspect if we did choose to use that dummy plate, that we would have to take longer in terms of making sure that reagents have been primed through before we started dispensing.
Okay. Another question from Robert at Boehringer. Do you use valve types with filters, or do you filter your reagents?
So I think the majority of our valves are actually without filters, and we routinely filter all our reagents through 0.22 micron filter, so that's all the buffers. Some users actually choose also to actually filter their complete reagents, so with their enzyme or their substrate, but our routine recommendation is that we would always filter the buffer component before adding your enzyme or substrate or whatever, your protein reagent. And some of our users choose also to use the suction filter cups that can be applied to the end of the tube. I think it's a bit more of a crude filter, but they choose also to use those as well. I think because with the number of plates we running, the risk, especially when you're automating it, you don't actually watch the dispensing, which might be taking over sort of 4 or 5 hours. The issue of having a blocked valve and losing it is obviously critical. So we try to minimise that by filtering as much as possible. Okay,
we have another question. Helen, do you do any cell-based assays?
Yes, and some of the data I've shown you there was from cell-based assays. So we've used them for cell plating, and actually see no difference with them in terms of reagent. We've not particularly met any problems. I think where you can potentially run into issues is more when you've got a very high proteinaceous solution. So if you've got cells in a very rich medium, I could envisage that you might have issues there. And as I said, most of the work that we do with cells for imaging assays is 384. So we would use the bigger valves, 0.3, 0.1s for those. That's our standard one. But obviously you can't use those on the 1536 well plates. So a lot of the data I've sort of shown you there, and I think on the automation platforms is more sort of biochemical. They're more of a workhorse for that. But we have used them for cell assays.
Yeah, and I think you've just answered that question. Another one of our questions which came in was, are you seeding cells in 384 and 1536 with the CERTUS? So
so we do seed them in 1536. We use the smaller valves, but that's for things like a reporter assay or something.
Okay. We've got time for another 2 questions. Lots of questions are coming in. We've got a question from Pia. Have you dispensed Matrigel with the CERTUS?
No.
Okay,
Sorry.
so that's fine. But answering that, Pia, we do have other customers, other users who have used the CERTUS FLEX for dispensing mattress gel. And if you would like more information on that, you know, we can share that with you. We've got time for one more question from Karen again. Do you only use gravimetric method for verifying your valves when using multiple valves for one dispense step? Do you verify the valves one by one or simultaneously?
So we have tried both. So I wasn't sure who asked the question, but I know the guys at Pivot Park, they've helped us over the years. They've actually supplied us with some sort of custom-made CAD printed devices, so we can actually calibrate, so we can verify, generate the verification factor by dispensing into tubes at the same time. We didn't see much difference whether we did that or just doing each valve one by one. So at the moment, we routinely do it one by one at the moment.
Okay, great. Okay, we do have lots of other questions, but we'll go through them after the presentation, and we will answer everyone's questions afterwards. Any questions specifically for Helen, you know, we'll send them across. Our details are on this last page here as well, so you can contact us at sales@fgyger.ch. Also, my email address, contact details, and Helen has kindly agreed to, you know, any of your questions, or if you'd like to get in touch with her for anything else. So once again, thank you all. A huge thanks for Helen and your team for everything that you've done for us. And yeah, I hope you all have a lovely evening and the rest of the day. And let us know if you need any more information from us. Thank you all. Thank you. Bye-bye.
Thanks, Sarah.
Thanks. Bye-bye.
Thanks, Helen. Thanks, Sarah.